And I would like first to thank the organizers, Professor Benjamin Shaare and Professor David Krashenov, for giving me the opportunity to present published and unpublished data here, and also for the opportunity to visit for the first time lovely Mexico. Great place. Enjoyed it very much. So there will be a little bit of shift. It is now shifting towards cancer and immunotherapy. It's precision cancer medicine and novel innate immune checkpoint and methods to predict patient's response. However, whether it's precision or personalized, I like always to say is that personalized cancer medicine is when you are trying to find a treatment or maybe a diagnostic protocol for a patient. Precision is when it works, actually. And not in all of the cases, it works. So the content would be some, this is a diverse audience, so there's some introduction about natural killer cells, natural cytotoxicity receptors, then about isoforms of natural cytotoxicity receptors, and about anti-membrane PCNA antibody that we developed for therapy of this immune checkpoint that I'm going to describe. But the ones that are in area, I'm going to give more or less what you might be understood at the end of this presentation. So we defined an innate immune checkpoint, which is between the NK express receptor, NKp44, and the proliferating cellular nuclear antigens that can also express on the membrane of tumor cells. And for these immune checkpoints, NKp44 PCNA, we have shown that NKp44 splice variant number 1, isoform 1, mediates this inhibition by this immune checkpoint of NK function, and that we developed an antibody against the tumor cell membrane associated PCNA that by binding to the PCNA on the membrane of tumor cells increase the NK function against the cancer. So it's kind of a treatment for this immune checkpoint. So these would be the conclusions that I will repeat them at the end of this presentation. So, slight introduction to natural killer cells. They belong to the innate immune branch, not to the adaptive, although they are large granular lymphocytes, but they do not belong to the adaptive immune response like T cells and B cells. And basically their function outcome is based on the balance between inhibitory and activating receptors that they have on the membrane or on the membrane of the NK cells. Here in the picture we can see actually in the green NK cells that is giving some kind of a medical massage to these cancer cells that eventually following this medical treatment, the cancer cells is dead, the NK cells is free to go and look for other clients. So we are going to discuss NK cells, and NK cells, as I said, have a broad spectrum of activating receptors that some of them, only activation of one of them can be, or some of them can be enough to mediate the function of the NK cells. I'm not going to list here, of course, the inhibitory receptors, that's a field by itself, but from this list of human NK activating receptors, we are concentrating in my lab on the natural cytotoxicity receptors, NCRs in short, particularly on the NKp44. Why NCRs? Because these receptors, in contrast to the others, are unique in expression to natural killer cells and to innate leukocyte cells, while some of the other receptors can be found also on T cells. So we mostly work on the NKp44, and a few years ago, before that, we did try to identify ligands that are recognized by these natural cytotoxicity receptors. We did identify viral ligands that are expressed on target cells upon infection with the virus, and these viral ligands are serving for the NK cells to recognize the viral infected cells and lyse these viral infected cells. And we did identify a paransulfate proteoglycans expressed or overexpressed on cancer cells are ligands as ligands for the NCRs, helping or co-mediating the lysis of tumor cells by the NK cells. But a few years ago, we actually identified the PCNA, the proliferating cellular nuclear antigen. It's a major protein in the virulence or in the function of tumor cells. And most of the work done on this protein, or really most of the work, is regarding its function in DNA repair and cell cycle in the nucleus. We have identified that this PCNA can go to the membrane of the cancer cell. And on this membrane, it can interact with the NKp44 receptor on NK cells And this interaction, although the NKp44 is an activation receptor, actually deliver inhibitory signal to the function not just of the NKp44, but to the function of all of the NK cells. So this we have identified a few years ago. And while trying to establish the system, we studied why actually the NKp44, a major activation receptor, can actually deliver inhibition to the NK function. And we realized that this is the only NK, or this is actually the only immune activation receptor that also have ITIM, immunotyrosine inhibitory motif in its cytoplasmic tail, that until our studies was considered as an inert domain for this receptor. We have shown that actually the interaction with PCNA on the tumor cells activate this domain through, of course, the tyrosine here, which is the amino acid that going through phosphorylation in this domain. And we have shown it by actually either deleting the ITIM domain or modifying, mutating just the tyrosine in this domain, and showing that following this mutation or deletion, the inhibitory function delivered by PCNA interaction with NKp44 is disappearing. So, looking on that, we realized also that NKp44 by itself has splice variants. This is not a surprise. Near 80% of our genes has splice variants, isoforms in the protein level, splice variants in the RNA level. And the splice variants number 1 of the NKp44 is having the ITIM, while splice variants number 2 and 3 of the same NKp44 do not have the ITIM. So, according to our theory, this variant should deliver the inhibition to the NK cells following interaction with PCNA because it does have the ITIM, the immunotyrosine-based inhibitory motif. But these two variants should not deliver inhibition. So, after realizing the issue of the isoforms, we indeed went to study that more carefully. First thing, of course, we generated the 3 isoforms of the NKp44 as is and transfused them into NK cells that do not express the native NKp44. And then we studied whether these 3 variants are interacting or not interacting with the PCNA. And indeed, only splice variant number 1 was delivering inhibitory function here, interferon gamma secretion, but also in the lysis level, to the function of NK cells following interaction with PCNA on the membrane of tumor cells. The other 2 variants did not deliver any inhibitory signal to the NK cells following interaction with PCNA. Here, this is a mechanism that we showed it in the level of the generation of the NKs, the natural killer inhibitory, the natural killer